Seth Markowitz and Reed Courtney, here presenting tonight on behalf of Team Silver, which stands for Studying Ion Ligands to Verify Endocellular Responses. It's not working. All right. Our preliminary research shows that over 70% of nosocomial infections, meaning bacterial infections that are contracted during a hospital stay in the United States, are resistant to one or more traditional antibiotic. This is a huge problem because antibiotic-resistant bacterial infections are very difficult to treat and in some cases can be fatal. One of the most common nosocomial infections in the U.S. is MRSA, which stands for methicillin-resistant Staphylococcus aureus. The chart on the below shows the total number of patient discharges in the United States that have contracted MRSA while in the hospital. Now, as you can see, from 1993 to 2005, that number has increased drastically. As we continue to use antibiotics to treat bacterial infections, antibiotic-resistant strains of bacteria will become more prevalent and can even create superbugs that you hear about on the news that are resistant to more than one type of antibiotic. So what can we do to fix this problem? We're exploring an alternative pathway that exploits an existing bacterial pathway. We're hoping by doing this, the bacteria will have a slower development of resistance to this new treatment. And additionally, we're looking for something that is toxic to bacterial cells and not mammalian cells because we want to kill what is making you sick and not you. So how will we do it? So iron is important for metabolic functions for many organisms on this planet. In humans, it's incorporated into the protein heme, which is in your hemoglobin, and bacteria use it for other proteins that are essential for life. When a bacteria is in an iron-deficient environment, it produces these large molecules called siderophores, which is on the right of the screen. Siderophores are bacteria-specific iron transport that go out into the environment and see iron-3, and they chelate to it, which means just binds very strongly to it, and then it brings the iron back into the bacteria to be used in those essential life proteins. Like I said before, they are bacteria-specific. That's very generally speaking. There are similarities between siderophore usage in bacteria in the same genus, but very generally speaking, it's bacteria-specific. And another important factor is siderophores cannot be taken up by mammalian cells. Mammalian cells lack the cellular machinery to interact with these siderophores and cannot get whatever they're carrying. In conjunction with siderophores, we also looked at metal genotoxicity. So metals such as silver have been used as antiseptics for centuries. Ancient Greek kings during wartime would carry around their food and water for very long periods of time and preserve it with different forms of silver due to its antiseptic properties. Another metal that is shown to have antiseptic properties is gallium. In some studies that we have looked at, it is proven to be genotoxic. So when it is introduced to a bacterial cell, it will actually interact with the DNA and disrupt the DNA and kill the cell. And gallium can also bind to siderophores. Gallium has a similar atomic radius and similar oxidation states to iron, and therefore it can bind to it when there is no iron in the environment, because siderophores bind preferentially to iron, but it can bind to gallium. So essentially what we were looking into is creating a siderophore gallium complex that can be taken into the cell. The bacterial cell will then use the gallium in place of iron, and it will disrupt the essential proteins that are necessary for life and cause the cell to die. So metaphor that we like to use at Team Silver is the Trojan horse metaphor. So I'm sure many of you have heard about the Trojan War and how the Greeks used the Trojan horse to trick the Trojans and let them into the city of Troy to destroy the city. So in this metaphor, our Trojan horse would be our siderophore, which in our case is Staphylococcus aureus. The Greek soldiers in this instance are our ion gallium, which will go into the city and destroy the city. And then our city of interest would be Staphylococcus epidermidis, which is the bacteria that we are studying. For more on the methodology, here is Seth Markowitz.
Our research project is a three-stage process. Our first one is a two-part. Our first step is to grow and measure bacterial cultures, in this case, Staph epi. This is important because we need to have the best growth we can in order to produce the most siderophores. The second step would be to quantify said siderophore production. We are using an assay, which I'll talk about later. Upon completion of those two steps, we move on to the second stage, which is forming the Staphylococcus A gallium complex. I'll call it SAGA from now on. This, again, is important because if we cannot bind the gallium to the staph, then it's not going to work as a treatment. And upon completing this, we'll move on to the final stage of our project, which is testing the treatment on bacteria. Over the summer, we started doing some research on Staph epi, specifically using a growth curve. In this case, we worked with different magnitudes of salt added to the media to test its effect. As you can see, there was not really much of a difference, but it's useful to point out what we're looking for. So many know there are 4 stages to a growth curve. The lag phase, which is the initial phase where there's the bacteria you initially have in the media. The exponential phase or growth phase, where as the name suggests, it grows at an exponential rate. The stationary phase or plateau, where you reach your final concentration of cells. And eventually the death phase, where they start to die off after the nutrients have been consumed. Specifically, our team is looking at the exponential phase to try and grow our staph epi as fast as possible. The faster we grow it, the more tests we can run and the more bacteria we can produce. And the stationary phase, because the more cells we have, the more siderophores and the greater chance of success. Specifically with the staph epi, we've been working with something called tris minimal media, or TMM. TMM is as I said, is minimal media, so there's not much in it. And we chose this because it, one, lets us choose the fuel source, so we could play around and try and get the staph to grow as fast as possible. And 2, it lets us limit iron. Now, as we said before, siderophores are emitted by staph epi to gather iron, and by taking out the iron, the bacteria will produce more siderophores in a desperate effort to get what little there is. So by doing so, limiting the iron, we are able to hopefully get the staph to produce more siderophores. Our first attempt was using mannitol as a carbon fuel source for the bacteria, and as you can see, we had an optical density, or OD, of about 0.35. It means there is growth, but there wasn't that much, and it's not really useful to us at that stage. So we moved on to a different fuel source called biotin. Again, similar growth around 0.35, which is not enough for us to really detect any siderophore production. So next, we tried cas amino acids in our most recent attempt. As you can see, this was our previous best, and now we're at around 0.6. So we almost doubled our production in our current research. But we're still working with different medias and different fuel sources in an effort to get it higher, because 0.6 is still iffy whether or not we'll be able to detect the siderophore production. As I said, the second part of our first stage is detecting whether or not we have siderophores produced. We're using something called a chrome azul S or CAS assay. In this test, what it does is the CAS is red normally color, and when it interacts with free iron, it turns blue. So, sorry about that. So in this case, as you can see, this graph's kind of backwards. You want to go left. As you go left, we have iron going away. In this case, we use EDTA, which functions similar to a siderophore, in that it picks up free iron and then binds to it and holds it and won't release. So as we get rid of the iron, the CAS begins to turn red. So we're using OD as red, and as the siderophore, EDTA picks up the iron, we have less and less red light emitted, and it sort of blue. All right, so once we've shown that this test is successful, and we hope to replicate siderophores. So the next step is to move on to the staphyloperin A gallium complex. In this case, once we know that the staphyloperin A is produced by the bacteria and we can measure it, we plan on isolating it through running through centrifuge. This will spin the bacteria down into a pellet, which we can remove and leave us with the fluid called the supernatant. And the supernatant will contain the siderophore, and we'll run it through rotary evaporation and other purification methods in order to make sure that we are only left with the siderophore in the liquid. And once we have said siderophore compound, we'll put it with gallium-3 ions, most likely gallium trichloride or gallium nitrate. And when we run this, we're going to use mass spectrometry to test whether or not we've actually binded it successfully. And hopefully we do, and we get to stage 3, which is treatment. So in this stage, we're going to run our test with the gallium staphyloperin complex. We're also going to run it with gallium salts to test whether or not it's just the gallium and not the siderophore bringing it in, and compare it to current antibiotics like penicillin and methicillin in order to test whether or not it is as or more effective than current treatments. And now, if we succeed at this, which hopefully we will, we'll move on to future research, which Reed Portney will talk about now.
Next step. So given where we are in our research right now, you may ask, how does what we've done so far apply to our initial problem of infections in hospitals? So the first thing you see is Staphylococcus aureus, and this is the bacteria that's responsible for most of the infections. We've begun working with Staphylococcus aureus and growing it, and our ultimate goal is to apply what we've learned about siderophore production by working with Staphylococcus epidermidis 2 aureus. The next thing you see is using different metals. So we discussed the virtues of gallium and how it can be useful as a metal in rendering bacteria non-viable, but there are lots of other metals that have similar effects on bacteria: copper, silver, zinc, and we would like to find out, or a future researcher could expand on our research, how do different combinations and concentrations of these metals affect bacteria. The next thing you see is siderophore and metal with an antibiotic. You may be familiar with the treatments that are used for Staphylococcus aureus right now. They call it MRSA sometimes because it's methicillin resistant. Methicillin is one of the antibiotics that used. There's also vancomycin, which is an even more aggressive antibiotic. So in conjunction with a metal, these antibiotics could be much stronger and could potentially work very well to eradicate bacteria. That's another future research goal we could look at. Ultimately, what a researcher would want to look at is manufacturing a working treatment using a combination of these things, the different concentrations of metals and antibiotics, to create something that's very strong and aggressively kills bacteria. Okay. We're going to finish off tonight by discussing our Gemstone experience, what we've learned so far with a few years of Gemstone under our belt, and we'll end with some advice for the freshmen and sophomore students that are here tonight. So our team composition, we have 12 members, and I think we're pretty evenly divided among different majors, though we definitely have a focus in people who are science majors, chemical engineering, that sort of thing. We currently have 4 sub-teams, one team examining the CAS assay, one team focusing on bacterial growth, one team focusing on developing new medias to better grow the bacteria, and one team getting our funding through grants. We've had different sub-teams in the past, and we've had members of the team on a variety of different sub-teams, and we found that it's useful to be very flexible in trying to contribute to the team in any way you can. So even if you're not familiar with a certain subject area, volunteering to be on a sub-team and learning a new topic. Some challenges we've run into. Early on, our project method changed considerably, and not a day would go by in the lab that we wouldn't have to modify what we needed to do. Our results are not always what we expect, even now. You run into unexpected problems. There's low growth. Things don't grow as you expect them to. Things like media that can be difficult to put together when there's a lot of different ingredients and they're very complicated. There's a steep learning curve for laboratory work. Not all of us are familiar with lab work. We have a few people who definitely focus in that area and do it for their classes, but we're definitely, by all means, we're not all very experienced in that area, and we learn every day that we go into the lab. A final challenge is arranging lab times and group meetings. For our sub-teams, for example, if, say, there are 4 members on the sub-team, how can we make sure that we all get together at the same time to work on media development or growing bacteria? So, some advice for students here tonight. First of all, try to coordinate your class schedules. This means not only just coordinating your schedules throughout the semester, like are you available to meet at this time, and okay, but also at the beginning of the semester when you're scheduling your classes. If you're in the same major, try to schedule for the same sections so that you'll be available after class to go and work together on your project. Holding your team members accountable is very important. This doesn't necessarily mean punishing anyone, but definitely making sure everybody contributes equally to the team in the way that they are best able to. It's important to get an early start on funding. The Gemstone program provides a lot of methods early on for you to apply for grants that are available specifically for Gemstone, and there are a lot of companies that have small amounts of money available for projects like this that students run, and those are all worth applying for. It's important to utilize your mentor and other resources that you have, like your librarian. Your mentor is somebody who has a lot of experience in the area you're working in and has probably done considerably more advanced projects in the same area. So you may save yourself hours of research just by asking them a simple question. And finally, don't be afraid to discuss your grievances with your group. If there's something that you're kind of bottling up inside or that's getting in the way of you completing your work successfully, your team needs to know about it so that you can move past it and get on to actually doing your project. Okay, so that's all we have. I'll take any questions at this time.